Very good. Well, good afternoon, everyone, and welcome to another one of our TESS seminars. TESS, of course, is our Center for Tropical, Environmental, and Sustainability Science here at JCU. Welcome back to a new trimester. So this is our beginning of another run, I think 11 or 12 talks that will take us up through the end of the year. This is my last run. I've been running or co-running TESS for 16 years, and Susan Lawrence will be taking over TESS next year. So no doubt be much better than it is now. It's a real pleasure to introduce Mike Kearney as our speaker today. We really appreciate Mike coming up from Melbourne and flying all the way up here to spend some time with us. Mike is a very well-known and very well-established ecologist and evolutionary biologist. He was trained at Monash University for his undergraduate work. He did his PhD at the University of Sydney, obviously one of the top universities, both of those among the top universities in Australia. He's now at Melbourne, one of the other top universities in Australia. So he's got fantastic academic credentials. He's been a Fulbright scholar in the US and done a number of other things I can't even quite keep track of. One of which is winning an Australian Laureate Fellowship, which is huge, really big. So he automatically gets to say, my name is Mike Kearney and I'm famous from now on. So anyway, yeah. Anyway, with real pleasure, please join me in asking Mike to speak to us today. Thank you.
Yeah, thank you very much for having me. I really love coming up to far north Queensland. I've been coming up here since, I think I was about 11 or so on a family holiday to Dunk Island. This weekend I'm going to go back there as a little nostalgia trip actually. So I've got, the wet tropics have been in my blood for a long time. And yeah, what I want to talk to you today is about the concept of the ecological niche and a way to model it that's possibly unfamiliar. It's a little bit difficult, but it's really rewarding. I have always wanted to be an ecologist, and I didn't think I needed any maths or physics really to be an ecologist, so I didn't do any. And then I had this sort of revelation when I was doing my honors research that there's a way to apply physical principles to understand the ecological niche. And it leads to a kind of modeling that links very directly to natural history. And to do it well, you have to have a combination of understanding the physical principles and concepts, but also really understanding the natural history, because this is a kind of modeling that works at the level of individuals and what they do all day. So maybe some of what I talk about might sound a little bit heavy. It kind of is. I've found it very difficult over the years to assimilate, but it's rewarding. And as I say, you have to be a good natural historian to do this well. And I hope that there are more people who try and bring together that combination of interests into this field of ecology. And it's a big puzzle trying to understand why species occur, where they do, and when they do. I don't know how many people in the audience have seen that book there, The Distribution and Abundance of Animals by Andrew Arthur and Birch. John Endler's nodding his head. That was a classic written by a couple of Australian biologists in 1954. But yeah, it's since they wrote that book, we still have trouble predicting distribution and abundance. There are a lot of pieces to the puzzle, and so you can see that there are many biological processes on the left here, spanning various scales. And obviously, we don't go down to these very short time and spatial scales at the quantum and molecular, but we do go from the cellular up to the organismal, up to the species distribution, and we have the interaction with other species. And then all of this is happening in the context of the physical processes that occur on Earth at different scales. How do we go about this? And there is this concept, the ecological niche, which has been used to try and approach this problem. So what I'm going to do in this talk is first just talk about the niche concept, and then introduce to you this physically based perspective of the niche, a thermodynamic kind of view of the niche. And then I'll give you 3 example applications, a couple involving lizards, one involving a bird. And then depending on how much time I have, I'll talk a bit more about the software that I've been developing to try and make this easier for us to do. So thinking about the ecological niche concept. So we have this a species of interest. This is a species that I work on, a grasshopper that you get in semi-arid Australia that's interesting because it's all female and clones, and it's just one clone. And that you can't really see the map of Australia very well there, but you can see, maybe peer carefully, you can see the range of the species is quite large, and that's the ecological niche mapped onto a landscape of one clone, one genotype. But So say one approach is to describe the environmental conditions that the organism experiences where it occurs. So this grasshopper is a very simple animal. It lives on a bush as a grasshopper, eating the leaves, and then lays its eggs in the ground. But we might characterize the thermal environments that the organism is associated with in different stages of their life cycle, and we might be able to describe which ones are suitable and which ones are not. And we might do the same for humidity, we might do the same for rainfall, and we end up with what Hutchinson came up with as the n-dimensional hypervolume perspective of the niche, which really just means you are delimiting the limits of the environmental tolerance of a species on multiple dimensions to n dimensions. And then you have this characterization of those environmental conditions, which you can then map to the geographic space, map to the landscape. And so that's what people tend to think of as niche modeling. And in particular, this kind of correlative niche modeling that is really, really widespread now, really powerful, where so things like MaxEnt, this kind of typical species distribution modeling approach, where the starting point are the occurrence records. It's the thing that you're actually interested in, the outcome, the distribution. And you link those to environmental data sets with some kind of a model, and then you project that environmental space onto the landscape. So start with occurrence, we have a statistical model, and then we end up with probability of occurrence as our output. So that's the typical way we think about modeling ecological niches. But this is powerful because it's process implicit. There are lots of processes that are being captured in the relationship between the occurrence records and the environmental data sets, which is what's powerful about it. But you can't unpack your model and find out what processes did I discover. And for me, that's unsatisfying because I want to understand the processes, but it's also limiting in terms of your management options if you don't know what the limiting processes are. But as I say, it's very powerful. So it has a lot of powerful uses, but it's hard to, there are limits because you can't understand what the processes are. So the other approach is to instead start with the organism itself and somehow link the traits of the organism that you can measure to environmental conditions through some kind of process explicit model. And instead of mapping probability of occurrence, you're mapping the outcome of that process. Did it succeed or fail? So that's quite a different perspective on the niche, and it's being process explicit. But there, as I mentioned at the beginning, there are so many different processes that we can consider. But if we can make a model of the ecological niche that's process explicit, we have an advantage because with the correlative approach, what we're doing is making an induction of where else those environmental conditions might occur. And if we are predicting to novel conditions, then we have to make an extrapolation, which is limiting. So not only can we not understand what the processes are, not only are we not able to have management levers from understanding those processes or have the scientific satisfaction of understanding them, but it also limits us to inferring only to conditions within the model development bounds and not outside. If we can develop a mechanistic approach that's process explicit, then we can make deductions and get around and make predictions to novel situations that we're encountering more and more as the climate changes and as species have been encountering as they've been moved around the planet through species introductions. So there's a lot to be gained if we can come up with a useful way of capturing these processes explicitly, but there are so many processes to look at, and a good place to start, therefore, is really fundamental processes. And that's why I'm talking about these thermodynamic processes, because thermodynamics is omnipresent, whereas other processes come and go. So this is where I get quite general. It's given away on the book here. This is Erwin Schrödinger, very famous quantum physicist. When he'd finished his work on quantum physics, he thought he'd turn his mind to something he thought was a more difficult problem, which is defining what life is. And he took a physical approach to this. What is the difference between living things evolving in this primordial slime and the things, the objects surrounding it? Erwin Schrödinger talked about the life in terms of physical principles of entropy, of thinking of the organism as a physical system, and very first thing that happened in the origin of life is a formation of a boundary. So we end up with an inside and an outside. So the cell membrane is critical. And in thermodynamics, what you're dealing with are state variables of energy and mass, and the processes of mass exchange through heat exchange and work. And we can think of physical systems this way, but we can also think of biological systems this way. So on the left, we have a military dragon. This is a dragon that I photographed at New Haven Wildlife Sanctuary on a moderately warm day in March, and it just run under a bush, and that's what it looks like in visible light. And then this is what it looks like in infrared. And it's in the process of exchanging heat and water. You can see its eye, if you look carefully. That's because there's water evaporating from that eye, and so it's cool. So you can almost see the mass exchange happening there, and also the heat exchange. It's warmer than its surroundings. It's moved into the shade of this bush. So we can treat the organism as a thermodynamic system, and we can draw a boundary around it. And that's what you do in thermodynamics. You draw a boundary around something you call the system. You've got the surroundings, and then you follow the flows of energy and matter in and out of that system. And the powerful thing we have in treating a system like this is the first law of thermodynamics and the second law of thermodynamics. So the first law being that energy and mass are conserved. So this is the equation for the change in energy, which is equal to any work done by the system on the surroundings, by any change in heat, and by exchange of matter, which has energy in it as well. But we also have this notion of entropy, which is what Erwin Schrödinger was really focusing on in his book. And entropy is a tricky thing to define, but it's essentially the dispersedness of a system, how dispersed a system is. So molecules are always jiggling around with their thermal energy, and as they jiggle around, they tend to disperse. And Erwin Schrödinger talked about organisms as being non-equilibrium steady state systems. So they're maintaining a state that is not in that direction that entropy would take it. They're staying steady at a state that is out of equilibrium. And he called that negentropy. And so you can think of entropy. The change in entropy is to do with the change in heat plus entropy production plus exchange of the entropy in any mass. And you can link the equation for the conservation of energy and the second law of thermodynamics equation for entropy exchange into this equation here, which the technical terms on the left is the entropy production. And then we have that is equal to the work the system does, the Gibbs energy, and the chemical potential. But those we can think of more biologically as the metabolic rate on the left. So that's the entropy production. The higher the metabolic rate, the more the organism is out of steady state thermodynamically. Work is movement. It gives energy changes the growth, and the final term on the right is food. And so this equation, it says what we all know, that in order to keep in that organized state out of thermodynamic equilibrium, an organism has to take in the energy from food. And then if it's going to grow, it's going to need more food. And if it's going to do any work on the system, it's going to need more food again. So there is a way to think of organisms formally as a thermodynamic system, and we can characterize it with an equation like this. And that is the goal of characterizing the niche physically as a thermodynamic system. Is it possible to do this? Well, it has been possible to do this for quite some time. The theory to do this has existed in various books, especially these ones. And if we're going to achieve this, we need to be able to characterize the environments around organisms. So it's microclimatology is required. And then the field of biophysical ecology is about the exchange of energy and or heat and water of organisms. That's been a well-established field since the 70s. And then metabolic theory, like the dynamic energy budget theory, that's also been around since at least the 80s. And what these books are dealing with are the really fundamental processes of temperature exchange, gas exchange, of water exchange, of feeding, all of the things that would become very central to your mind if you were suddenly left on your own in the bush, in the desert, up on a high mountain, the actual fundamental things that we need to survive. So these are all ecological things that we can think about, but we can actually formalize them from a physical point of view. And the processes of temperature, of heat exchange, of respiration, of water exchange, and of feeding are actually quite linked in a very particular way, in the way that these words have just been rearranged. And I'm going to show you in a bit more gory detail exactly what this looks like and what the terms are, so you can see how the connections exist between these processes that reduce the degrees of freedom of the system, so that if you stick these processes together into a model, the model's got less wriggle room, and you end up being able to make quite powerful predictions, more than you might expect, more than I'm used to with the sort of ecological models that I was dealing with prior to learning about this. So the first equation, this is an energy balance equation. So all of these are basically equations obeying the first law of thermodynamics, that the energy and mass must be conserved. It's for temperature. So we've got various processes by which heat is exchanged in this equation. We've got exchange by solar radiation. We've got exchange by infrared radiation coming in. So all the surfaces in this room are emitting infrared radiation, and we're all emitting infrared radiation. Then there's the metabolic heat being produced by the organism. That is that term of aliveness that I showed before, the entropy production. And then there's heat being lost from the surface of the organism as infrared radiation. There's convective exchange, depending on the air temperature and the wind speed. There's evaporation, and there's conduction. So all those processes, except for solar radiation, are occurring for us right now in this room. And then connected to this heat balance, you can see there's two terms here, the metabolic term and the evaporation term, where there's this dashed circle. This is where you have a coupling between energy and mass exchange. So for mass exchange with the metabolism, we have this, when the metabolism's running, it's running based on the food that's being consumed. So heat's being released as that food is burned. So food comes in, some of it is released, the energy is released as heat, and then some of the mass goes into growth, reproduction, storage, or feces. And then also coupled to this metabolic term, we have oxygen being consumed in the process of metabolism, and then carbon dioxide, nitrogenous waste, and metabolic water being produced. And then finally, we have connecting with the evaporation term. So evaporation is a loss of heat, but it's also a loss of mass because water is being lost. So some water is being lost here. Water is coming in from any food, any water in the food. There's also that metabolic water. It connects here with the respiration term here, with the metabolic water coming in as part of the water. So we're all getting some water from our food right now. So that input must equal the water coming out in the urine, in the feces, what's evaporated, and what's stored. And you can see that the food demand from the metabolic system is also driving how much water is coming in. So if we know how much food they're eating, we also know how much water they're getting. They may drink extra water. If we know how much nitrogenous waste they're producing, then we know how much water they must be losing in their urine. And if we know how much fecal matter they're producing, then we know how much water is lost in their feces. So you can see the whole thing is very tightly connected. I don't know, I found that formidable when I first saw it. It's really just adding stuff. It's just various terms being added together. Every single one of those terms involves functional traits. So there are some terms about the organism in each term, like the solar radiation term needs to know the solar absorptivity of the organism, needs to know the surface area. And every single one of those terms has some functional environment as well, like maybe the wind speeds needed, or the air temperature, or the solar radiation. So that's the goal, to try and characterize an organism in this way. And what I'm going to do for the rest of the talk is show you some examples of how this is possible, how this has been done. And as I mentioned, the theory exists to do this. So for understanding heat budgets, we have biophysical ecology and microclimatology. And understanding the microclimates is actually half of the challenge here, because you have to get the environment that the organism is experiencing. You can't to solve these equations. So, and then the metabolic terms, what's now highlighted, are all dealt with by the dynamic energy budget theory. And I guess the main thing I've contributed to this field is reading that book, Dynamic Energy Budget Theory, figuring out what it does, and then connecting it with the other two books basically, and sticking it all together. And then when you have that, you're able to simulate the trajectory of an organism starting as an egg or an embryo, going through its whole life cycle, through some gauntlet of environmental conditions, solving these equations and saying yes or no, did it get through? And if it got through, then that set of conditions is part of the ecological niche of that species. Right? That's the concept. So I don't know how abstract that sounded, but let me try and make this sound a little bit more concrete with some examples. And if you do find this interesting, a whole bunch of us that work in this field got together and wrote an overview of the state of play of the field of biophysical ecology in global change biology. And that should give you an entry point into the literature. And as I'll mention at the end, there's also the NicheMapR package, which does all of these calculations in R. And for those of you coming to the workshop, we'll go into some more detail with the NicheMapR package. Okay, so some examples. First one, this was a really fun project. Started a while back. Eric Pianka, if some of you have heard of Eric Pianka, he's recently passed away, and he didn't live to see this study published, but he collected tons of data, field data on reptiles in the Great Victoria Desert in Australia from the 60s, and in the Kalahari, and also in the US. And we were talking about, well, to what extent could we predict some of the observations you made, because he measured the body temperature of every lizard he caught, and he measured the air temperature, and also their behavior. There's a whole book summarizing his life's work, Ecology and Natural History of Desert Lizards, and you can see the tables on the right show the years he did his collecting and how many individuals he collected, and they were all put into museums, and so tons and tons of data. But I was able to get access to all of his field notes and all of the raw data a while back. Now it's actually available on a website, but so we started playing around with this data, and here's an example of one of his field notebooks. So you can see he's got the specimen ID. I think that links to the specimen in the museum. The species, whose name has now changed, that is the military dragon. And then the sex and the age. He's got the air temperature and the body temperature. So he just had a thermometer which he held at Eric Pianka height, and then he shoved that thermometer into the lizard and got its body temperature. We've got the time. We've got some hieroglyphics that indicate the pattern of behavior with which the lizard, he was mostly interested in its escape behavior, but this also tells us whether they're in the sun or in the shade. So really useful for seeing whether we can predict what they're doing with these models. So here's the modeling pipeline that we developed. It's the general way that we do this kind of modeling, where we need some climate forcing data, some environmental data that's like the weather, like what a weather station would measure. There were no weather stations at the Great Victoria Desert or in the Kalahari where he was doing his work, but there's this fantastic product called the ERA5 product, which is an hourly, back to 1950 gridded data set of it's basically reanalysis data, which is running a general circulation model, highly calibrated on observation. So it's a complete data set every hour of everything we could possibly want as forcing variables for these models, which we downscale to the habitat level with the microclimate modeling. So we can put the slope and the aspect, the substrate, absorptivity, all those sorts of things, and be specific about the kind of habitats the organism's in, like what the conditions would be in sun versus shade. We need the traits of the organism to feed into that model, things like the temperatures at which they forage, their body mass, and that sort of thing. And then we feed that in to make calculations of things like the body temperature, could they be active, how much energy would they need. And I'm not going to say too much about that study other than just to show you how well this kind of modeling approach can work. So here's an example of an entry of Eric Pianka's field notes plotted on a map. So this is, I forget what day this is, but you can see the body temperature predicted by the model is this line here. And the dots are where we expect them to be active. these vertical bars are when it's, the vertical bars are how much shade they selected. So they're in the shade in a triodia bush at night, and then they're predicted to come out at about 6 in the morning. And then you can see that the body temperature is quite flat because the model is selecting shade as the lizard requires it to stay within its thermal requirements. And then at night time, you can see it finishes foraging. And the red dots are Eric Pianka's measurements of body temperature through time. And the arrows are pointing at his observations of when they're basking, when they're in open sun and in the shade. And then he didn't collect in the middle of the day when the lizards would all have been expected to be hiding in the shade because it's too hot. So you can see it's giving you, this is 1967 or something, in the middle of the Great Victoria Desert. This is with a global product of environmental data through a microclimate model predicting the hourly behavior of a lizard back then. When we look at all the air temperature observations and body temperatures, air temperatures in blue, body temperatures in red, that he measured for military dragons, they were all points he measured for this one species. We get within a couple of degrees of the temperatures of the animals. So it can work really well when you bring all that physics together to predict the organism's body temperatures, but also the biology of their behavior and habitat use. Okay, there's a lizard example. And with a lizard, what we're doing is solving the heat budget for what body temperature balances the heat budget. We can also do this for birds or mammals, endotherms. But instead, we know what the core temperature is generally. We're trying to maintain a particular core temperature, and we can ask what metabolic rate must you have to be at that temperature. And if that metabolic rate ends up, like the model will say, it might say in a hot environment that the metabolic rate needs to be below what is possible, what the minimum is for an organism, it might even be negative. Then it says, okay, here's how much heat you have to dump by evaporation. So the model will tell us for an ectotherm what the temperature is, from which we can work out a series of things like whether it can be active and what its metabolic rate would be. For an endotherm, we can predict how high the metabolic rate would need to be in the cold, and we can predict how much water they would need to dump in the heat. So we were interested in doing this for a very charismatic bird, the night parrot, which I'm sure you all know about. Steve Murphy was working on this bird out at Pullen and interested in their water requirements, how often they would need to go to a dam. There was a dam nearby to get water, or whether they could get their water from their food. This is the habitat in which they live. During the day, they're a nocturnal bird, of course, night parrot. They're living in a big trumpet triodia, so the same habitat as the military dragon, in fact, and dealing with the heat. And they can't drink, they're just stuck in that bush. And then during the evening, they go out onto these plains, grassy plains, where they're feeding on various grasses, grass seeds. But there's also this sclerolina, kind of like a kinopod. Well, not kinopod, but it's a spiky but very succulent plant. And there was a video here of Steve crunching one, and there was a lot of liquid in there. He was wondering, he'd found evidence that they were hanging around this, and he was wondering, if they were eating this, maybe they don't need to go to water so much. So our goal was to do the calculation of how much water would this bird be needing to thermoregulate in the bushes during the day, and could it actually persist without going to water? So we had to model the microclimate of this bird. My thermal camera has taken a picture of a night parrot. That's as close as I've gotten to a night parrot. I actually caught up with Steve Murphy. It's actually at a tree kangaroo workshop at Roger Martin's house up in Malanda. And we were talking, he was about to go in the field. He said, we were going to do this study together. And he said, what can I measure? And I said, take my infrared camera. He took that image, which we used then to, he's got very cold hands. I think he was nervous with holding this bird. But we used this to test some of the predictions of the model. But we also put lots of loggers out to see what the temperatures were in the habitats that they're living in. This is just a reminder. We make this calculation of the heat and water and the nutritional budget of the bird. We had to also, well, another way to test out the model was to see how well we can predict the metabolic rates of a bird in a metabolic chamber, where you put the animal in a chamber and you change the temperature and you see what the metabolic rate does, what the water loss rate does, and what the body temperature does. We didn't do this for night parrots. We did this for budgerigars. made sure it worked for a budgie, and then turned that budgie into a fat budgie, basically, to make a night parrot. But it worked well for the budgie. So the top one, you can see metabolic rate having to be high in the cold for the budgie until about high 20s. And then they feel comfortable. They're in their thermoneutral zone. They're comfortable in this, which is more like low 20s. And then as it gets hotter, their metabolic rate starts to rise. And that's partly because they've let their core go up, because they don't defend their normal resting core. They'll let it go up a bit. That saves them water, but it's stressful. And you can see also the water loss rate going up. The black is the model predictions, the red are the data. So you can see the model can make this prediction for the energy and water requirements of the animal in the lab. And so then we see whether we can do it in the wild. But you always have to check to see whether or not you're calculating the microclimates right. So this is a plot of time series of temperatures under one of those triodia hummocks. And you can see it gets pretty hot, even though those are quite shady bushes. The dashed line, I think, is about 45 degrees. And you can see the red are the observations of the temperature of this lie button in that clump. But you can see the black are the model predictions. And we can get really close to the conditions in those bushes in the field. But this is, if they were just sitting, this is a logger just sitting 4 centimeters above the ground in the complete open. Look how hot it's getting, like way over 60. So they're definitely doing better sitting in that triodia bush. But yeah, we also incorporated the food budget and the water budget. So we worked out how much water was in the feces, and then the composition of the food, which helps us get to that metabolic water production. So we could work out how much water they were getting for free from their food. And then we tried 3 different water contents as if they were eating relatively dry seeds or the sclerulina bushes, which we estimated to be between 25 and 55% water. So here's an example output of the model. So we've got for a full year, this is the microclimatic calculations showing the surface temperature, the air temperature at 4 centimeters, and the air temperature where the parrot is in its bush or at night where it's foraging. So you can see that dashed line of about 45 degrees, the parrots are experiencing in our simulations quite high temperatures, but they're buffered by being in those bushes as compared to out on the surface. And they see the blue vertical lines are rainfall events. So we're out near Longreach, some big rains and then not very much. This plot shows the core temperature of the bird in red, the skin temperature of the bird, and then the outer plumage temperature. You can see that they're out at night foraging, the plumage temperature is quite low, around 10 degrees. But you can see during the summer, their core is being pushed up, so they're letting their core go up to, because it's hot, but that saves them from having to dump too much water. But nonetheless, this is how much water they're having to dump, according to the model, grams of water per hour. So we can say, well, how much water would that be per day? We can say how much dehydration would you have per day under these different diets. We know how much water they were having to lose. We know how much water they'd be getting from their food under the different diets, and they're the different colors. So the water-rich diet in blue, and then the low water diet in red and then orange in between. And so where it's above 0, it means they're dehydrating. Where it's below 0, they're getting a profit in water. And then we can accumulate that, and we're also, when it rains, allowing them to drink. And that's what you see on the right. And that's, so we've got percent desiccated, what percent of their normal body mass have they lost? And the 2 dashed lines are the bounds within which we think birds die. So when they're between 11% and 22%, they're the numbers in the literature, they're lethal. So when they're feeding on the sclerulina, they're okay. They don't get to that lethal desiccation level of 11%. But you can see if they're on a seed diet only, they're going to have to go and drink from that pond, that dam, which has a predation risk, of course. This is looking at it. I mean, the power of having these models is you can then generalize to other places and other times. So we can go back in time and say, all the way back to 1996, how dehydrated would they have gotten in the past? So you can see good years and bad years from this point of view. And so this, how often did they get to that 11% dehydrated? with the 3 different diets, and then the 22% dehydrated, not too bad. This is the cumulative dehydration. So they are getting quite dehydrated if they're not feeding on the sclerulinas. And that's under the current climate. Then we can say what would happen under the 2070 climate. And I mean, the thing that I've noticed with a lot of work in this area where we're doing physiological studies of temperature effects, it's very thresholdy. And once you get up above, like it don't have to go too much higher, and then things get really bad because basically you're hitting that physiological limit. So you can see there's the 2070 climate that we simulated here, even with spirulina, they're frequently dehydrating at the 11% level and the 22% level, so not good. But interestingly, you know, they're the places that we know the night parrots to occur in a triodia clump or out in the open plain. But what if they went down into a hole, just a little bit under the ground? What if there was like a little burrow in the triodia that they could go into? That's what we had before with the nasty 2070 climate. This is if they go down a hole. Massive effect. This is something we don't know about. Do night parrots go down holes? We don't know, but some birds do. Okay, one last example, which is the sleepy lizard. I presume you've all seen this lizard before. It's a pretty common lizard in southern Australia. It's very charismatic. And we know a lot about it because of the person pictured there, Mike Bull. So like Eric Pianka, dedicated his life to field biology, and he focused on just one species and studied them for 40 years. He was initially wanting to study the ticks on these lizards, studying a hybrid zone, not really interested in the lizards, but fell in love with the lizards. He found that they fall in love with each other for life. He found they're monogamous, with one couple coming together each year, each spring for 40 years. We had a really, really wonderful long-term data set that we could use to test out these models with, and including this contraption, which he called the waddleometer. So that lizard you can see there on the wandering across an open field has this is like, obviously the technology's got better these days, but this was back in I guess in the 90s. It's a device that has a step counter. It's like a little magnet that counts every single step the lizard takes, and it's got a thermometer and it's got a light reader. And they also had GPS tracking on these lizards. So we could not only look at their body temperatures, but we could also see how fast they were moving. So here's the example set of data for all of the lizards they have. So every spring they would go out, catch lizards, put the loggers on them, change the batteries each week, every 2 weeks, I think, and get really detailed data on body temperatures. And that's the data set for 2009 and 2010. You can see from September through to December. And then the black dots are what we predicted with the model for the same time period. And again, this is not using local weather station data. It's using a continent-wide product. And then in this case, we went sort of the whole hog and applied the dynamic energy budget theory, which I'm not going to go into too much detail about, but it's extremely powerful and really hard to understand. But once you get it, it's really powerful to do that whole life cycle modeling of the animal. And like it basically fills in this stuff here, like I mentioned before. The night parrot, we did a bit more of a shortcut approach, but this is using the full dynamic energy budget theory. And it was really interesting. So this sort of somewhat complicated looking plot shows for that time window from September to December, it shows for each day, for each hour of the day, lizard activity observed and predicted by the model. There's grey dots here. Those are the nighttime hours. So you can see the day length increasing as you go into the summer. The light blue squares are hours when we would predict they would be thermally suitable for the lizard to run around. So in the early time of the year, you can see there's white space here. That's because in the early morning and the late afternoon, it's too cold for a sleepy lizard, and so they're not moving. And then as you get into the summer, you can see some white patches in the middle of the day. That's because it's too hot for a sleepy lizard to be on the move in the middle of the day. And then the red dots are male 11885. The size of that square is the number of steps it took each hour from the step counter. So you can see pretty good correspondence. There's no lizard activity in this hole where it's too hot for lizards predicted by the model. Lizards weren't active then, and in all these white areas here, they weren't active. And this prediction of these blue dots are predictions for a lizard that's being active every single hour that was thermally suitable, right? But lizards aren't always active when it's thermally suitable. This next plot is of individual 11533. And so you can see there's not much red there. So this is a female, and this was typical of the females. They hardly moved at all. The blue dots are when the dynamic energy budget model said they'd be hungry, right? So this is the other side of the constraint. There's what's thermally suitable, and then there's what is necessary. When do you need to come out to replenish your food supplies? So the females are moving as little as possible. The males are running around all the time. That's because they're mate guarding females. So we can capture those dynamics with the models. You can also see these blue spikes are rainfall events, and the dashed lines are predictions of how desiccated the animals would be. And this ended up being very interesting. They look like a pretty tough lizard, but water is important for them, we discovered. So these two plots show recapture rates, fractions of lizards recaptured. Because remember I said, during the field work, they go out and catch their lizards at the beginning of spring, and then they have to keep catching them again and again to change the batteries, right? So they're usually getting about 80% of them each time. But during 2009, you can see a drop in the number of capture rates. In the model, what we saw during that gap, in the data, what we saw during that gap were some flatlining body temperatures. So you can see the red line, so that's this gap here. The body temperatures have flatlined. So any idea what that might be due to? That's a wombat burrow in the picture. They're living in southern hairy-nosed wombat country. They're going down burrows. And this blue line, remember, this is how desiccated they are, and they get to about 15% desiccated by the model, and that's when they seem to be just saying, right, this is too dry, we're shutting up shop. And in fact, this is why they only do their field work to December, because they can't find any lizards after December, because it's just too dry to find them. Down that wombat burrow, it's going to be almost 100% humid, so they're not going to get any food, but they can wait down there for the water. So when we simulate them to do that, when they become desiccated, then our predictions of their body temperatures match. Hooray! But and here's the equivalent plot I showed you before of the activity pattern. So again, you can see they're not being active here. But individual 12434 seems like a super lizard, because it's active through that whole time period. And this was puzzling. At this point, when we noticed this, we hadn't looked at the GPS data. I'd just been working with the time series data and not looked at what was happening in space. But then when we looked at what was happening in space, so they had lots of data on home ranges of lizards. So this is a view of the site on Google Earth and all the home ranges of the lizards before the dry spell. Then for the ones that were active during the dry spell, the home ranges were clustering around a feature on the landscape. That feature was a water body. So the lizards that had access to that water were not shutting down. That's Mike Bull sitting by the dam. And so, yeah, really, really clearly showed from that really wonderful data that Mike had, but combined with this modeling to get the inference that water is really limiting these lizards. And so, so if we were to run a trajectory simulation of one of these lizards with the dynamic energy budget model, so doing all that churn of all those calculations and those complicated equations through time, the top graph shows the growth trajectory. The bottom is, you can see the rainfall levels in blue, but you can see if they had access to drinking water, the growth trajectory, this is just their body mass, they're growing up, and then these drops here are when they have a clutch of say, live bearers when they give birth. As opposed to when they don't have access to drinking water, you end up with the graph at the bottom. So when you run that calculation across the whole continent and you say, what is a lifetime reproduction? it gets better and better as you go further and further north because it's, it's getting warmer and the generation time's faster and they can have more babies. But when you incorporate dryness, then you start to lose, the reproduction starts to go down in the center, and that starts to come closer to the species distribution. And I show this also as an example of how you can end up making a map with this kind of analysis. What I showed at the beginning with the whole MaxEnt approach, you start with a map with distribution points and you produce a map. You can't really do that easily with this approach. What you're doing is, as you've seen, trying to find limiting processes, and you can map those processes onto the landscape. You might get lucky and have identified the correct ones. And so it does seem that part of the story here as to why the sleepy lizard's distribution looks the way it does is not because of thermal limits, it's because of water. So hopefully those examples make it a bit more concrete how these models work, how they tie into natural history, basically things we can measure about what individuals are doing all day. And now I'll just finish by telling you a little bit about the tools that I've been putting together with my colleagues to make this easier to do for ecologists. Certainly it's things I've done to make it easier for me, and then it also makes it easier for others. So the NicheMapR package, the idea is you're mapping the niche to the environment, to the landscape, characterizing it and mapping it. And it has a whole lot of functions in it, a whole lot of microclimate functions that are basically connecting this one microclimate model in the package to different data sources, including that ERA5 one that connects to there's one that connects to the silo data now for Australia. So there's a whole lot of different functions that are basically allowing you to use different sources of weather data. You can also use your own weather data. Then there's the ectotherm models, which are solving the heat budget for the body temperature of the organism. But recently, I've added in a version that calculates leaf temperature and has the, you can specify the stomatal density and behavior. And the microclimate model also has a full soil plant atmosphere continuum model with stomatal dynamics as a function of soil water potential too. So if that's of interest, I can tell you more about that on offline. There's an egg model, which is basically a model that calculates the uptake of water from the soil according to water potential drivers. There's the endotherm model. There's a very simple one called the ellipsoid model, which is really good for teaching. And then there's the more sophisticated ones, what we use for the night parrot. And then most recently, I've made a human one, which is in review, but basically just sticks together an ellipsoid with a few cylinders, and you can put clothes on it. It's fun. And then there's the dynamic energy budget theory, so there's functions for them. And perhaps most useful for if you're interested in this and want to get started, and this is what we'll be doing in the workshop, is playing around with some Shiny apps, which Shiny is just a way of making web applications to interface with R packages or R software that allow you to just play around with these models without having to write any code, and I actually quite like using them because it's just quicker. So there are apps that run the microclimate models with different environmental data sets. There's a whole list of them on this page, which you can just find by Googling. It's not too hard to find. And most recently, I've made one that links together all the models. So the dynamic energy budget model with the ectotherm model and the microclimate model. So this is getting towards the dream where you have this database of the traits, truly functional traits in the model that you need. So you can choose a species, in this case, Tenotus pantheritis, one of Eric Bianca's study subjects. You choose that species. When you do that, it pulls out the dynamic energy budget theory parameters from a big database that has 6,000 species in it. But we haven't yet got all the other traits that we need for the heat budget. We're hoping to do that down the track. But you can put in your location, the start date, the data set you want to use, which stage you want it to start at as an egg. You can say how deep in the soil it is. And then there's lots of parameters that you can change, and out comes a trajectory of wet mass through time for a year. And you can also see, so that this plot shows starting as an egg in the ground, the mass is sort of slowly declining, and then hatches. The brown is actually food in the stomach. And then the pink is the reproduction buffer filling up. It hadn't quite gotten to maturity to lay an egg by the end. It's got plots of the body temperature and whether the organism was active. This just shows in a bit more zoomed in that trajectory of mass with the egg developing, hatching, food. And then you can see this is the temperature of the egg in the soil, and then it hatches and starts running around and thermoregulating. You can make the plots that I showed you of the activity windows. This lizard can be nocturnal, so you can see I've allowed this animal to be active into the nighttime, which are the blue, dark blue here. And you can do things like I showed you with the sleepy lizard, where you can say, I want to give it complete access to water, which is the left-hand side, or I want to make it only get water from its food and when it rains, which is the right-hand side. So this way you could play with these models and see how they work and get a feel for them and see whether you can be bothered trying to learn how to use them, because it is a steep learning curve. So I said at the start of this talk that there are lots of pieces to this puzzle of understanding the distribution and abundance of organisms. What I think this modeling approach that I've described allows you to do is get the edge pieces of the puzzle. That's how I like to do a jigsaw, right? It's bounding the problem. And there's all that complexity inside the jigsaw of interactions with other species. But the thermodynamic principles give you the edges. And so in terms of energy, in terms of heat exchange, energy in terms of food, mass in terms of water. So hopefully I might have inspired some of you to go down this path, and I'll see some of you in the workshop to inspire you further. Thank you.
Thank you very much, Mike. Good audience. Questions? Now, the host is always supposed to have a question ready to go for these particular moments. I did want to ask you one thing about the night parrot. You said there was a particular shrub, which I don't remember, spirulina. Okay, and is that affected by grazing and land use and things like that? Or I'm just wondering if that's so critical for its distribution, is that something that's being affected by environmental change, or is it just fine and happy?
I don't think they're browse. I don't know for sure. They're quite spiky. You certainly see it commonly in Arizona habitats. So I suspect it's not that heavily browsed. It's not like we know for sure they're eating those. Maybe Steve Murphy has learned more about this. It was more like he did some tracking and he found them at sites that had a lot of this around. So it's quite speculative, but so I don't know for sure whether they're even eating the spirulina. So I can't answer your question very well.
Okay. But it does seem to be very important for their thermal regulation.
Well,
is that right?
It just shows you how important the water availability in the diet can be.
Right. Yep.
Yeah. I'm going to ask a question. Then we'll go to people online. My boss just put up a hand, so I'm just getting in before. You mentioned something about if they let their core go higher to avoid losing water. So rainforest species, they're living in an environment where water is generally not limiting, and they wouldn't normally be. So I'm thinking inverse of that. So they would not normally be able to let their body temperature, they're not adapted to letting their body temperature go higher in order to avoid losing water, because they're not normally in a situation where they avoid losing water. So that means that they're more at risk of heat stress or dehydration. What would?
Yeah, I mean, the dry season, a long time ago, I did something on radar gliders. It was like my first adventure modeling. And that was with some guidance from Andrew Crockenberger, and he was telling me how in September, October it is quite dry. So there is a time when they can be limited in the wet tropics. But I think when we're trying to set up these models, you do have to set up what is their sequence of thermoregulatory responses. And yeah, you would expect, say, a water bird or some freshwater bird, animals that do have access to all that water would just not let their core go up. But as you say, like if they're caught out in a situation where water is limiting, they may not have the capacity to tolerate those high temperatures. That's where you're getting at, right? Yeah. So it's true.
So does that mean that you would expect them to die from heat before they die of dehydration? and then vice versa. So some things will die of dehydration maybe before heat.
Sometimes the heat's too much to get, like the capacity to dump the heat by evaporation is not sufficient. So we have a certain maximum sweating rate, and our acclimation response is to increase that. So athletes can sweat 3 liters an hour. I think a normal person is like under a liter. So there is a physiological limit to dumping heat that by evaporation that may not, meaning that you might end up, even though you covered yourself in water, there's enough power of evaporation to cool you. Because obviously as it gets more humid, then that capacity goes down too. Right? So hot and humid is bad.
We can't hear his mic sensors.
Oh, okay. Should I use this label mic?
Oh, I think I have.
Oh, yeah, that's why. Is it if I do this, it might be better.
That's good.
Yeah.
Thank you.
Yep, no problem. Yeah. So yes, sometimes the limiting factor is dumping the heat by evaporation. Sometimes, even with that capacity, you get too hot because there's no capacity to dump the heat because it's too humid.
Okay. Other questions online? Or yeah, we have an online question. Sorry, just wait a second. Go ahead, Lynn.
Can't hear you, Lynn.
Thanks very much for the talk. It was interesting. I was wondering if it's dangerous to collaborate with you. You killed 2/3 of your collaborators. You don't have to answer that. No, what I wanted to ask you is, do you think you can scale this up in some way to like population numbers? Because like we were working on, or a long time ago, we were trapping regularly in a place where there was lots of Tenedos pantherines sometimes, and sometimes there wasn't very many. And it just made me think about this, like what if we were losing everything out of the landscape that wasn't close to water because they just didn't survive? And other times, or maybe they just moved closer to water and then they moved out again. So I guess I was wondering about population consequences of the kinds of models that your individuals do. What would that mean about how they use the landscape?
Well, yeah, when I last saw you at the herpetology meeting, Lynn, I then went to Canberra for a workshop on how to link these models to population dynamics. And these models give you vital rates because, you get the time to maturity, you get the number of offspring produced, and you can get survival from entities from stress. And you can also link activity levels with mortality too, because the more active things are, the more vulnerable they are. So yes, it can be done. It hasn't been done very, very much, but there's a person in the audience, Alejandro, who did this for wet tropics possums. So that's a really good example, just came out in Global Change Biology recently, if you want to see how that can be done.
Okay, thanks.
Now, Michael, I've got a question. That's just here. Back to the night parrot and the dehydration. You mentioned there's a band between 11 and 22% dehydration that's lethal. Why is there an upper limit?
No, sorry, it's more that, the experiments to work out lethal dehydration, it's hard to do, right? So people don't do them. So it's rough. Like, this is anecdotal evidence of birds dying at 11%. Sometimes they can survive 22. So it's like a range which birds have been observed to die. Does that make sense? So it's not like there's an upper and lower limit. It's more that, it's just that it's somewhere in there is probably where their limit is. Yeah, ours is actually really low. It's like more like 7 or 8. There's a certain point, I think it might be 7 or 8% when we can't drink anymore because our mouth is like swollen. So yeah, the 11% is a lot.
Okay. Any other questions? Lucas?
Thanks, Mike. That was super interesting. So all the parameters that you have to put in to make those calculations, I guess that you get them the best way that you can.
Yeah.
But do you find, or do you have an expectation that there's plasticity in those parameters? Would you expect that kind of acclimation that would influence the predicted outcomes?
Yeah, and this is basically ecophysiology. It's applied ecophysiology, and so
Yeah, and you can incorporate those sort of responses, right? So, I mean, the most complicated application I've done so far in that sense is the plague locust egg. It's highly plastic. They lay eggs that are diapausing or non-diapausing. They have quiescent phases, depending on the photoperiod and the water, soil moisture levels. So, you can do rough models where you're putting in rough numbers, and often that will tell you whether or not to go further, whether you're getting close to a limit or not. But then, yeah, things are complicated. There are these plastic responses. There's subtle behaviors. So the way to think about these models is they're a great kind of comparison for your empirical work, and where you find a mismatch is exciting because they're either you've made a mistake or there's something interesting that they're doing, especially if the model says no and they're still, and they're there. They've got some trick up their sleeve. One example, which wasn't quite like this, but it was a trick up the sleeve of an animal that we discovered through the course of doing this work, was a past student of mine, Nat Briscoe, was studying koalas, and she noticed them flaking out on the tree trunks, and we were measuring all the things you need to measure for the microclimates, and we decided need to measure the tree trunks, and noticed the tree trunks were a lot cooler than the air because they've got water coursing up them, I guess, and they've got some inertia. So there's a trick that you might have calculated, no, koalas should be dead, but they're dumping heat into the tree. These are the sorts of things you might see as you do this kind of modeling and compare it with detailed field data.
Other questions online? I just have one thought. When you're talking about the fact that you seem to see a strong threshold oftentimes in physiological responses, and unhappy and then dead. It just struck me so similar to what they're saying with spectacled flying foxes and some of the other flying foxes here. 41 degrees, they're stressed and they're unhappy, but they're alive.
Yep.
42 degrees, they die en masse by the thousands.
Yep.
It's just such a striking thing. Is your sense that many species exhibit this kind of striking threshold with respect to temperature?
Yeah, like with endotherms, what's happening there is as it gets hotter, their core is going up, and then usually that leads to a higher metabolic rate, because metabolic rate goes up with temperature, which adds to the heat load. So it turns into a runaway process. So it very quickly spirals into a meltdown with an endotherm. Yeah. And then with ectotherms, the thermal performance curve is usually quite asymmetric looking. It sort of rises slowly as it gets hotter and then comes crashing down soon after you reach near the optimum. So the physiology is there's a cliff for endotherms and ectotherms. And so a small change in air temperature can tip them over. And with the modeling, you do +2 and everything's fine, and +4 and it's, whoa, that is really, really bad. It's a bit sobering.
Yeah, it sure is. Any other questions? Mike, we also really appreciate, I forgot to mention, but we really appreciate you coming and offering this lab, the special course that you're offering here. That's wonderful for our people. So we really appreciate it. And please join me in thanking Mike Kearney for a fantastic talk. Thank you, Mike, for coming.